Hello, my name is Eve Marder, and I'm head of the Division of Science at Brandeis University and a professor in the biology department. I'm also a neuroscientist, and for many years I've been interested in trying to understand how the behavior of circuits or their dynamics depends on the interactions between neurons via synaptic connections. But before starting, what I would like to do is just pay homage to some of the very important people who've come before me and who have given rise to the field that I'm part of. In particular, I'd like to say that Peter Genning, Don Maynard, and Maurice Moulin were there in the very early days, and sadly they're no longer with us, and I really thank them for their contributions to understanding small rhythmic circuits. Also, I'd like to thank Al Selverson for his prescience in understanding the usefulness of the stomatogastric nervous system, the preparation I've studied for my entire scientific career, to understand many, many different problems in neuroscience. And I would also like to pay homage to all of the workers who have studied the stomatogastric nervous system now for more than 40 years. They're too numerous to mention, and everything I'm talking about today basically builds on the work of hundreds of people working for many, many years. But what we're going to be talking about today, and you see my title there, Understanding Circuit Dynamics, Lessons from Small Rhythmic Circuits, Variability, Compensation, Modulation, and Homeostasis. And I've divided this presentation into 4 short parts. In the first part, I'll do an introduction to central pattern generators, or CPGs as we call them, and I'll explain that in a moment. The next section will be about neuromodulation, which you know is an important facet of how the brain works, found in all animals. The third will be about homeostasis in the brain and nervous system. And then the fourth is a topic we've been studying today, most recently, which is about animal and animal variability and all sorts of properties. And what I hope you'll get from this combined set of 4 talks or 4 modules is an understanding how it has been possible to use a nervous system, a small nervous system. It contains several hundred neurons in totality, with a ganglion, the stomatogastric ganglion, which only has 30 neurons, how it's possible to have come up with many fundamental general principles relevant to all nervous systems, whether they be humans or snails or worms or flies, by studying basically the properties of 30 neurons. And those will be the tales I'll tell you. I'll try and let you understand how much important how many general principles we've been able to extract from studying the same set of neurons over and over again. So to start, module number 1, an introduction to central pattern generating circuits, or CPGs as we call. Now, rhythmic movements such as walking or walking sideways or walking sideways in the other direction are produced by central oscillatory circuits, in this case in my spinal cord, but for many animals in various and sundry ensembles. And these circuits have been studied, or central pattern generating circuits have been studied in a variety of animals, including leeches, lampreys, flies, snails, frogs, mice, and chicks. But one of the most useful preparations has historically been the crustacean stomatogastric nervous system. So next I'm going to show you a little bit about the stomatogastric nervous system. So first of all, I'd like to show you on the top, you see a lobster. And what Elizabeth Rezer did when she made these recordings is she drilled a hole in the carapace of the animal and stuck wires inside the hole, and then pasted everything back together again, and then released the animal back in the tank. And then she was able to record electromyograms, or EMGs, from the behaving animal as it went about its business. And those EMGs are seen here on the bottom, and what you see is a triphasic motor pattern where we've labeled LP, PY, and PD, and that triphasic motor pattern continues. In this case, this is the pyloric rhythm, and it continues throughout the lifetime of the animal. Now, Elizabeth also was able to show that this rhythm changed in frequency and in some of the other of its attributes or phase relationships according to the behavioral state of the animal. But the important thing is that this rhythm, like your respiratory rhythms, are ongoing for as long as the animal is alive. Now, this shows you what the behavior is in the intact animal. Now, to make the preparations we routinely make in the laboratory, we study the nervous system in vitro because it's very difficult to record from the nervous system when it's covered up in a hard carapace or a shell. So we prefer to remove the nervous system, and what we do is we take the stomach out of the animal, and then we dissect the nervous system from the surface of the stomach, and then pin it flat in a Silgard-lined petri dish. Silgard is a clear plastomer, as you've seen here, and then we superfuse it with physiological saline that would have the same ionic composition as what the animal's hemolymph or blood would have. And we make two kinds of recordings, and you'll see these at different times throughout these talks. We record intracellularly with microelectrodes, seen here in green, red, and blue, from the cell bodies or somata of the stomatogastric ganglion or STG neurons. And we record extracellularly using wires placed. Actually, what we do today is we build a Vaseline well, we put a wire inside the well around the nerve, and then we record between the inside of the well and the outside of the well, or, as shown here, it can be done with two hooks on the nerve. But the extracellular recording gives us recordings of the action potential times, and now you can see a comparison of the in vivo recordings I showed you before on top and the in vitro recordings that we've made with intracellular electrodes on the top three traces and extracellular trace on the bottom traces from the isolated stomatogastric nervous system. Now you can see that the in vitro or the isolated nervous system is also producing that triphasic motor pattern. It's also LP-PY-PD. And the extracellular recording on the bottom shows different amplitude action potentials due to the activity of the LP-PY-PD neurons, and that's because the axons that run in the nerve at which this, from which these recordings were made have different sizes, and therefore the action potentials have different sizes. But in any case, what I want you to see here is the similarity between the in vivo recordings and the in vitro recordings. Now, in these in vitro recordings, we've thrown away the animal. There's no more animal. There's no more sensory feedback from the muscles. There's no more information from circulating hormones. There's no more sort of inputs from the brain or from the mouth parts. But this is an example now of why we call these a central pattern generating circuit, because this circuit in isolation continues to produce a fictive motor pattern which is almost identical to the motor patterns that were produced when the animal was alive and kicking. Now, this is sort of the proof that central pattern generating circuits actually exist. You can take the nervous system out of the animal and they continue to produce the motor patterns that they would have produced if the animal were still attached. Now, the stomatogastric nervous system has been so useful for understanding the mechanisms of rhythm production circuits precisely because the in vitro system works so well. So what can we next say? The first thing that Don Mader and Al Selverson set out to do almost 40 years ago was to discover the connectivity diagram among the neurons in the circuit to try and come up with a wiring diagram that would basically explain why the rhythm existed and why it had the particular pattern it has. So what people did is they recorded simultaneously from 2 or 3 neurons, and they looked for synaptic interactions. And on the basis of those synaptic interactions that they saw in pairwise recordings, it came up with a connectivity diagram, and that's what you see here on the right side of the slide. And in this connectivity diagram and all the others I will show you, the resistor symbol means an electrical synapse that's formed by gap junctions that allows electrical current to flow back and forth. And the black dots are chemical inhibitory synapses. And you can see here that this connectivity diagram includes a mixture of electrical coupling and chemical inhibitory synapses. So the first thing to remember in this particular circuit, and this is true in many rhythmic motor systems, that inhibition is extremely important for the generation of patterns. Now, the other thing I'd like to sort of step back and talk to you about for a moment is today in the world of neuroscience, there's a great push to try and come up with or describe what's called a connectome, or a wiring diagram for all sorts of animals. And those of us who've been working in small nervous systems for many years know, on the basis of a lot of history, that this connectivity diagram is absolutely necessary and completely insufficient to predict the dynamics. If I had given you this connectivity diagram or connectome ahead of time without showing you what these dynamics actually are, there's absolutely no way you would have been able to go from this connectivity diagram to these dynamics without a lot more information that's presently there. In 1980, we basically had this connectivity diagram or connectome. And what we've done in the years from 1980 until more recently, the large we, all those of us working in the stomatogastric nervous system, have tried to understand how this connectivity diagram gives rise to these dynamics. And I would just like to point out that this connectivity diagram or connectome is absolutely necessary and completely insufficient for explaining dynamics without a lot more information than you're seeing here. And what would that information be? What's missing in this connectivity diagram as you see it right now is you don't know the strength and the time course of all the synaptic connections, and you don't know the properties of all the voltage and time dependent channels that are found in each one of those cells. And without all that information and a way to put all that information back together again, you can't go from a connectome to dynamics. And so the large enterprise being pursued today in the world is a necessary start to really understanding brain dynamics in larger systems. Now, so this just summarizes what I just told you. The connectivity diagram or connectome is absolutely necessary and completely insufficient to explain circuit dynamics. But now we'd like to sort of continue with the stomatogastric nervous system and give you a few more pieces of how it works. On the left, there's a stick picture of how we make the recordings again. Now you see the Vaseline wells around the nerves and the intracellular recordings, but you also see three anterior ganglia, the two commissural ganglia, they're called the COGs, and the esophageal ganglion or OG. And those ganglia send projections, modulatory projections, down the stomatogastric nerve that you see there and into the stomatogastric ganglion. When all of those projections and modulatory inputs are left intact, what you see when you record is a more complicated set of recordings than I showed you before. You see not only the pyloric rhythm, which now on this time base you see as the oscillations in the PD cell, but you also see a slower gastric mill rhythm as alternations between activity in interneuron 1 and LG. And now you can see DG down here and GM here. And so this is now a slow rhythm, which is happening simultaneously with the fast rhythm. And I should say that the gastric mill rhythm moves some teeth inside the lobster stomach. The pyloric rhythm moves a filter on the back end of the stomach. So we have a pyloric rhythm, which is ongoing, always on in the animal, and a gastric mill rhythm, which is episodic. So that gastric mill rhythm is much more like your locomotor motor patterns, which you turn on and off under different behavioral contexts, and the pyloric rhythm is much more like your respiratory rhythms, which are ongoing and for as long as you're alive. Now, this is the full connectome of the stomatogastric nervous system. And over here we see the cells you saw before, the pyloric cells. And then over here you see the additional cells that are part of the gastric mill circuit. And what I think you can see is that there are a tremendous number of electrical synapses that are connecting neurons not only within a circuit but across circuits. And that turns out to be very, very, very important, because you can see that these electrical junctions basically connect many, many, many neurons in a weak syncytium that actually allows, as you'll see a little later, neurons to change their relative firing properties and give rise to sort of interesting challenges in understanding how these circuits work. But I'd like to go back to the pyloric rhythm for a moment and sort of take you through some of the questions that people asked about rhythmic circuits, not only in central pattern generating circuits, but rhythmic circuits as you find them in the brain. Those questions historically were, number one, what generates the rhythm? Why is the circuit rhythmic at all? And what determines the timing or the phase relationships of the rhythm? That is to say, why is the circuit rhythmic? And then what determines exactly the order and the timing of all the elements in the circuit? So, in terms of rhythm generation, there are two major ways in which you can imagine building an oscillator to power a rhythmic circuit. One way would be with bursting pacemaker neurons that would have properties similar to what you have in your heart. That is to say, cells that can depolarize and hyperpolarize rhythmically in the absence of inputs. And the other is that an oscillation can arise from circuit interactions. And I'll briefly show you both of those. So in the top here, we see a recording from a PD neuron. And you can see that it depolarizes. It goes up, it fires a burst of action potentials, and then it hyperpolarizes or becomes more negative, and then it depolarizes and goes more positive. And in this bottom panel, you see the same kind of behavior, but now in a model neuron where we've used differential equations to describe the properties of a number of different voltage and time-dependent channels to build a bursting neuron. And again, you can see the depolarization, the membrane potential goes more positive, it fires a burst of action potentials or spikes, and then hyperpolarizes, and the cycle repeats. Now, it turns out in the pyloric rhythm, the AB neuron is a bursting pacemaker neuron or an oscillator. It's electrically coupled to the PD neurons, and it's because of that electrical coupling that the AB and the two PD cells that you find in each animal are a bursting pacemaker kernel for the rhythm. So that's why the pyloric rhythm is rhythmic. But the other mechanism for generating a rhythm can result from interactions between cells, two or more cells, that are not themselves intrinsically bursting or oscillatory. So, and these kinds of circuits that I'll show you in a moment are often called half-center oscillators and can arise from reciprocal inhibition. So here you have two cells here which are not coupled, but we're going to couple them, and I'll show you this in a moment, with reciprocal inhibition. And also we're adding to the circuits a conductance we call an H conductance. This is a hyperpolarization activated inward current, which tends to make the cells depolarize or become more excitable after they've been inhibited. So let's see what happens when we make that circuit. Here are two GM cells that are both silent when they're uncoupled. And now we're coupling them up with reciprocal inhibition, and we get these beautiful alternating bursts between one and the second, one and the second. And what happens is when one cell fires, it inhibits the second cell, but then the second cell, because of this H current, it escapes from the inhibition. Eventually it crosses threshold, it starts firing, inhibits the other cell, and the cycle continues. So now this is what we would call an emergent oscillation, where the oscillation does not depend on the bursting pacemaker properties of a single neuron, but arises from synaptic connections. And this circuit configuration, reciprocal inhibition, is very, very, very common, not only in motor systems, but throughout the brain. Okay, the next question is, what determines the phase relationships of the relative timing of cells in a circuit? And I'd just like to again show you the pyloric rhythm again. Now these are extracellular recordings that are color-coded for your ease of seeing them. And again, you see the PD, LP, and PY. And we define the period as the time from the first spike, for example, in the PD cell, to the first spike in the next burst of the PD cell. And then we can ask, you can measure the latency to the off of the PD, right there. You can measure the latency from the beginning of the PD burst to the start of the LP burst. And then you can also measure the time to the end of the LP burst, and you can measure the times for a PY start and PY stop, or on or off. And then if you divide those times by the period, you can calculate a phase. That is to say, you're normalizing the on and off times to the period, so you're getting the relative timing of the elements in the circuit. But before showing you the phase, I'd just like to point out that in recordings from many, many animals, we get a quite large variation in the frequency of the pyloric rhythm recorded under the same set of circumstances. In contrast, the phase relationships that I just described to you how they're calculated are remarkably invariant. So even though there's tremendous variability in the frequency here periods going from about 0.4 or 0.5 seconds to 1.5 seconds, the phase relationships of the relative timing of those elements are invariant. Now, this is a question which, if you think about, turns out to be quite non-trivial to understand, because remember, phase is relative timing, and so we've turned properties which would have, you would think, fixed delays into constant phase. And just to explain why that's not trivial, I'd just like to show you here. Now, on these sort of phase diagrams, here's a triphasic rhythm with 1, 2, 3, 1, 2, 3. And now, if we just kept the time of number 1's burst and number 2's burst, their time's constant, but the period of the whole oscillation slowed down, then you'd see you get an altered pattern, because number 3 might fill up the pattern. If you did the opposite and you kept the time of activity of 1 and 2 constant, but now, as you see there, but now, because it's going so much faster, number 3 doesn't have any time to fire. In contrast, on the bottom, you can see network activity with perfect compensation. Here at this frequency, you've got 1, 2, 3, 1, 2, 3. And now when the network slows down, what happens is the time at which they're on changes so that the phase relationships are maintained. So you maintain the same pattern, although it's going much more slowly. And likewise over here, you see you can maintain the same pattern as it goes faster. And it turns out that it was quite a challenge to try and understand how this arises, because membrane currents and synaptic currents all would have fixed time constants. And so it turns out the answer, and this came from a lot of work from a lot of people, is that there are multiple processes that determine when, for example, the LP starts to fire after its inhibition, and that includes the time course of the synaptic inhibition. It includes currents such as IH, or the hyperpolarization activated inward current, or currents like a fast transient potassium current, and it's the interaction between multiple cellular processes that allow a central pattern generator to be phase constant. And so this, I think, is sort of an important insight, that determining something which must be very important for the animal, such as maintaining constant phase, actually depends on the interaction of many different membrane currents. Now I'd just like to show you one more slide about some of the things that can occur in the stomatogastric nervous system. And that is, there are neurons that can switch between firing in time with the fast pyloric rhythm and firing in time with the slower gastric mill rhythm. So on the left hand, you see intracellular recordings from the PD and the LG neuron. PD you've already learned to know and love. It's a member of the pyloric rhythm. LG was always described as part of the gastric mill cycle. But when the gastric rhythm is off, the LG neuron can fire in time with the pyloric rhythm, which is what you see here. Notice LG is firing in time with PD. But when the gastric rhythm turns on, LG now is firing in a long gastric mill burst. So these are neurons that can switch back and forth between firing in time with the fast rhythm and the slow rhythm. So they're coupling themselves between two different pattern generating circuits, and this kind of behavior almost certainly is important in your cortex as neurons couple up together in different ways in different functional ensembles important for behavior. Now, at this point, I would just like to acknowledge the people in my laboratory who've been involved over the years. I've been extremely privileged to have large numbers of wonderful people working in the lab. My present lab is over there on the left. Lab alumni. There are a large number of people who are now in independent positions of one form or another. And then I've also been tremendously lucky to have a very large number of very talented collaborators. So thank you very much.